Welcome everyone. Thank you for joining us for this recording. This isn't the recording that took place at the live event. This is a recording that we're doing after the fact. So we're going to try and make it a bit more interactive. Gary will be taking us through the slides, and in between that I might just have one or two questions for Gary. If I can just introduce Gary to you quickly. Gary is an anaesthetist. So he helps us at Insight with a lot of clinical work. So he's not trained in immunology specifically, not an expert there, but his clinical background will bring quite a bit of understanding to the topic. So he's going to take us through some of the basics of the immune response. And it's quite a complex process, the immune response, so he'll take us through all the different levels, and then we'll also talk a bit about vaccinations and testing and herd immunity. Gary, do you want to start us off and just start with some of the basics on the immune response?
Thanks for the introduction, Zelda. Yeah, so this is not the live event. It'll be a bit more conversational, I hope. And I'll start by saying that some things that everyone knows, I think, that SARS-CoV-2, which is the official name for the virus, is a new virus. It's causing a new disease, and it's led to massive uncertainty around the world, difficult policy decisions, disruption of healthcare systems and of the global economy. As we all know, the world, in a word, has become unbalanced or imbalanced. Fortunately, in South Africa, and certainly in Australia, I know this presentation is intended for an Australian and hopefully global audience as well. We've largely avoided overwhelming the healthcare system, which was the initial concern that we all shared. But along the way, over the last 6 to 8 months, unfortunately, as many as 30,000 South Africans have died from the disease, that's COVID-19, and in Australia, over 800 deaths as well. And along the way, we've learned a lot, but there are many questions, much uncertainty, and many challenges remain. So that's why I've sort of subtitled this Uncertainty and Imbalance. So as Griselda pointed out, I'm not an immunologist. The immune system has become a hot topic, and I think many people are interested in it and how it works. And I'm going to try and present some of the basic ideas around how the immune system responds to viruses, this virus in particular, and therefore even for a non-immunologist or non-clinician holds important insights for what has happened and what may happen in the future. And importantly, I think how to protect ourselves from the disease, from COVID-19, and how to understand the evolution of the disease in individuals and in the population as a whole. So the immune system, I've learned, having had to sort of refresh my basic knowledge of it, is arguably the most complex part of the human body outside the brain. And I'm going to summarize it in 2 or 3 slides. At least some of this will be correct, I hope, and some of you will be better off for it, and possibly better informed than certain world leaders, I think, who you know who they are. Barry, my colleague, and his modeling team in the Actuarial Society and so on, are hoping, I think, for more appropriate coefficients for their models. They will not get them today. And having explained a little bit about how the immune system works, I'm going to then address a few of the more interesting and perplexing aspects of COVID-19. So in the context of the biological response to the virus that causes it. The immune system enables us, as this graphic shows you, to live in a world full of competing life forms, bacteria, viruses, and other small chuchas. When healthy, we're oblivious to them, even though they live inside us and on us, on our skin. In fact, they're necessary for human health. For example, gut bacteria, that's the microbiome, actually trains our immune system early in life. And this complex system works remarkably well, but it can be subverted by cunningly evolved organisms. Occasionally, it goes out of control and damages or even kills the host, destroys the host. So over the last 8 or 9 months, an enormous amount of new knowledge has emerged around COVID. And many people have become experts or may think they're experts. I'm trying to avoid becoming, as Zapiro would have it on the left here, a Professor Karen, PhD in immunology and professor of everything from the University of Facebook. But the reality is that even experienced scientists are having to learn very quickly about this virus and about the underlying factors that have caused this tremendous impact on human health and on the global economy. So to the right is a reference to a website hosted by the National Library of Medicine in the United States, which is one of the best places to go for scientific publications. So as of August 30th, so that's the day before this live presentation, or the live version of this presentation, there were 46,500 publications. And I looked yesterday and there were nearly 52,000 publications around COVID-19. So just give you a sense of how difficult it is, even for experts, to keep up with all the new knowledge. Okay, so how does the immune system work in three slides? So we'll start with what's known as the innate immunity part of the immune system. So this is the part of the immune system that works relatively quickly, that is within a matter of hours, extending to 3 or 4 days. This part of the immune system is the dominant host response. And certain immune cells, such as macrophages, neutrophils, and others, respond to bacteria, viruses, other microorganisms. And part of that response is to generate special chemicals called cytokines or chemokines. Interferon is one of them. You may have heard of this. And these chemicals do 3 things, broadly speaking. They signal neighboring cells to put up barriers to infection. Secondly, they signal infected cells. So the virus may get into and start multiplying in certain host cells, but the innate immune system signals those infected cells to die. So hopefully not too many die, but our own cells are signaled to die by host immune cells. And then finally, the third aspect is that this part of the immune system recruits other white blood cells to stimulate a longer lasting immunity. And along with that, they generate inflammation. And inflammation is the characteristics that all of us are familiar with, redness and swelling and pain. So those are the characteristics that you may experience and see when you become infected. The innate immune system is a pattern recognition system. So it may not be able to identify specific organisms, a coronavirus or a specific bacteria, and it's thought of as a non-specific response. So it's not targeted on individual microorganisms. Nevertheless, is a very effective and important part of the immune response. So that's innate immunity, number 1. So I'm going to go now to the second major part of the immune system, which is called adaptive immunity. And this comes into play after a few days, so around the 3 or 4 day mark. It's the more specific part of the immune system targeting specific microorganisms. And it has a long-term, not only days, but months or years of effect to protect us from infection. So the key cells in the adaptive immunity response early on are these special little guys called dendritic cells. And so when viruses come in through a surface, so inside your throat or your nasal passages, these dendritic cells become infected and they then become messengers, and they go to certain other parts of the body, specifically the lymph nodes, and also to an extent in the spleen, which is like one giant lymph node. And they become educators, if you will, for these special white blood cells called lymphocytes. And there are two kinds of lymphocyte. There are the B cells and the T cells. And the B cells become specialized versions of themselves called plasma cells. And those plasma cells secrete antibodies. And those antibodies become important in attacking viruses and virus-infected cells. And similarly, the T cells do not secrete antibodies, but they play a critical role in addressing virus-infected cells. So there's a special education process that dendritic cells are responsible for, activating these B and T cells, or B and T lymphocytes. And this slide just shows you a little bit more detail about both the antibodies from the B cells and the T cell response. So you can see that there are two kinds of antibodies. There are so-called neutralizing antibodies. So as the term suggests, those are the ones that are key to stopping any infection of host cells. Then there are other non-neutralizing antibodies that help other white blood cells to become active in actually destroying host cells. And that's the graphic that I'll point to with my cursor here. And then there are other T cells that can directly, so without any need for antibodies to be active, there are certain T cells that can also become active. They recognize a viral protein, so here a viral antigen attached to a host cell, and can destroy those infected host cells. And these T cells, it's emerging, have a very important role to play in explaining whether populations are resistant or affected or even die from COVID-19. And then finally, this graphic top is the so-called spike protein, which in the very many illustrations or graphic representations of the virus are those little spiky things that the virus has. And that protein here is illustrated bound to an antibody.
Okay, so sorry, Gary, if I can just interrupt you there quickly. Maybe you can just explain what it means when we have these T cells that have started to recognize different viruses. Does that mean that we fight off the infection quicker, or we just have milder symptoms? Maybe some of your slides that you go through next can explain some of that. And then I don't know if you maybe just want to hazard a guess around the literature on T cells and this sort of innate immunity that we have, because that literature seemed to emerge a bit later through all of this understanding of COVID, and that actually has quite an impact on modeling and how we understand the virus, and in different populations, what sort of underlying levels of immunity they might have.
That's a great question, Griselda. I think you are becoming a professor of immunology along with me. And I'm going to try to deal with some of those issues in the subsequent slides. Everyone, I think not everyone, but many people, I think, had heard about antibodies as a concept that some people, at least some people, were familiar with, but far fewer people were aware of the idea or the existence of T cells. And I think what the literature is showing is that T cells T cells that are protecting us from the disease, from any infection or from more severe forms of infection, may well exist in the population. So I'll try and cover that in the subsequent slides. So the third, and I'm going to come back to sort of my train of thought here in terms of the third essential element of the immune system. And the remarkable thing about the immune system is that having been exposed to a particular organism, whether it's that exact same organism or something quite similar to it, our immune system gives us a memory of it going forward, so that a subsequent re-exposure to that organism or that germ will no longer result in disease. And that's the remarkable thing. So this graphic from a very recent publication, and we're going to make these slides available, I think, in some form, so people that are particularly interested can go and read this review. It's somewhat clinical, but very up to date, and is a 2 or 3 page explanation as of today or the last few days about COVID and the immune system. So what this shows is that initially the B cells, the ones that make the antibodies, arise within the first 2 weeks or so. It's at that point that almost everyone that is exposed to the virus produces antibodies. And, you know, with luck, those are neutralizing antibodies that are highly protective. But almost everyone, including those people who do get sick, produce antibodies. In fact, the sicker you are and the longer your illness with COVID, the amount of measurable antibody goes up and goes up over time. So this graphic shows the initial infection, and then the purple and the blue here, these plasmablasts that make the antibodies in response to the infection, and then quite soon the memory B cells, the ones that are going to hang around for some time at low levels, but are going to be around in order to respond to a subsequent re-exposure. So here is my arrow at the point which you might see the virus again, because it's not going to go away, but you'll have a response to it shown here in the dotted gray line. And these are the different kinds of antibodies that are produced. There are IgM, IgA, and IgG, which are special varieties of antibody. And then I'm going to show you the T cell response above. And the T cells have various flavors or varieties. There are CD4 cells and CD8 cells. And in South Africa, some of us will be aware of CD4s because they are what we measure in patients who have HIV infection to see how well they're doing and whether what's going on with their immune system. So CD4 cells and CD8 cells are part of the host response. And again, there are memory T cells, just as there are memory B cells, that if you're re-exposed to the virus, have a response and they multiply and protect you from getting the disease a second time. Okay, so that's the immune system in a nutshell. And I'm going to now tell you, or go into 9 or 10 different scenarios that all have to do with the immune system, and talk about the various aspects of the disease, who is susceptible, and so on. And Griselda, you may have a few more questions along the way. So really, this one is about 2 things. It's about the language that we use when we describe the immune system. So technically or clinically, you know, immunity is about response. So, which, as I've said already, is extremely complex. But I think in common parlance, the language, when we talk about being immune, we think of protection. We think of not getting the disease, not getting any symptoms, hopefully, and certainly not becoming ill or severely ill from the disease. But it's not as simple as that, and the immune system offers protection, but in various degrees and not absolutes. And we'll come to the question about reinfection in a second. So you can have something called sterilizing immunity, which is absolute immunity, and you may not have any symptoms, or you may have functional immunity. In other words, you are sort of protected from any significant illness, but may still have some symptoms. There's the concept of waning immunity. In other words, the immunity fades over time and may disappear completely and be lost. So those are some of the terms you'll hear in reference to the immune system. The next concept I want to come to is came up quite early in the epidemic, before it really hit South Africa, or Australia for that matter, which is whether certain populations, which were already, back in February, March, seemed to be more resistant than others to the infection, and people were wondering whether that was simply a question of getting different time trajectories and where the disease and its carriers went to first, or whether it was some aspect of the population that afforded them protection. And one of those factors that was thought to have a role, and may in fact have a role, is BCG vaccination. So BCG is a vaccine that is administered to most South Africans and in many other countries, but not others, that is somewhat protective against tuberculosis, which, as we all know, is a major problem in South Africa, and it hasn't gone away with COVID. So we certainly still have TB, and in fact may be up for much bigger problem with TB going forward than in the past. So the graphic on the left, in the red are countries that generally have very little or no BCG vaccination versus countries in blue where BCG vaccination is routine. And you can see, based on this graphic from a particular paper that was published actually by, including some South African authors, suggesting that maybe South Africa would be protected from a major problem with COVID. And the reason this may occur is because of another concept which is called trained immunity. And trained immunity is a property of the innate system, which we spoke of a few minutes ago. So that initial host response, the innate immunity. So BCG may condition certain innate cells so that they have a much better response to a secondary infection. And that has to do with some complicated stuff in terms of host DNA and the DNA that governs how your T cells actually respond. I think we still don't really know whether BCG vaccination is protective, but there's some suggestion that it does. The second thing I want to talk about is the critical question of how COVID-19 actually causes the damage that it does. The 30,000 or so deaths in South Africa and the many more in other parts of the world. So here there's this concept of a cytokine storm, and as we already said, cytokines are molecules secreted, produced by host cells, not by the virus itself, by host cells, T cells and other cells in response to the virus that cause all the damage. And this graphic shows you an alveolus, which is part of the lung, where oxygen is transferred across cell membranes and into the bloodstream and carried by hemoglobin in red blood cells to the tissues. And you can see here that the virus enters, that's step one. Macrophages are part of the innate system that I referred to. And the cytokines are those molecules, as we said, that draw in other immune system players. And then that cytokine storm, or overreaction, if you will, causes the production of certain other products. One of them is fibrin, which is kind of a sticky, gluey type of stuff that's involved in blood clotting, but excess production damages the alveolus, damages the blood vessels, fluid leaks out of the blood vessels into the lung and causes respiratory failure or lung failure. So the points A, B, C, D are the current ideas, I think, about how all of this causes the disease. So what people think currently is that the virus is evolved to cause a delayed innate response with less interferon, less of the early molecular signal. It somehow exhausts the T cells, so they're less active in doing the job that they need to do. And in effect, both the innate and the adaptive systems are compromised. More virus replication and migration of the virus to other parts of the body that it might not otherwise reach. So both a delayed response in terms of destroying the virus and a prolonged immune system activation actually causes the damage. So it's not so much the multiplication of the virus, but the host's immune system that causes the disease. And of course, this has implications for treatment. So the better we understand the evolution of the disease, the more likely we are to be able to come up with effective treatment. And really, at this point, the only drug that's shown to be really effective in reducing death from COVID is a steroid medicine that suppresses this immune response to a degree. Okay, the third thing I want to talk about as far as the sort of scenario is concerned is reinfection. So a couple of weeks ago, just before the live presentation, were a couple of documented cases that were fairly convincing of individuals that were reinfected. And of course, everyone is extremely anxious about the idea that COVID will not only disappear, but in fact will repeatedly infect us, causing huge problems over time for the foreseeable future. In this particular case, it was an individual who had a confirmed infection with the coronavirus, and then months later, 140 days or more later, had a confirmed reinfection. However, this individual did not become sick, was asymptomatic. One of the factors that may have been involved is a different strain of the virus. And so the questions around these reinfections are whether these are actually new infections or just a persistence of an existing infection, because if you do the tests for coronavirus, it takes a long time for the test to become negative, or whether the immunity that some people have is actually not sterilizing. So using the term that I referred you to in an earlier slide. So I'd say the consensus at the moment is that we don't have too much to worry about from reinfection. When you have millions of people that are already infected with coronavirus, I think we're going to see occasionally some people become reinfected, but probably not become severely ill. The next thing I want to talk about is the very well-known differences in predisposition to severe disease. And so we've known right from the beginning of the pandemic that older people are much more likely to become infected, become sick, and die. And there seemed to be a male predominance in most populations of people who are affected by the disease. And again, the differences here probably lie in the immune system. And this was a recent paper showing that at a cellular level, that their T cells were much more activated in women than in men, or females and males during SARS infection, COVID-2 infection, and this is sustained into old age, this female versus male and older age. And again, these immune system findings have implications for who needs the vaccine the most, and maybe even for who should get a second or even a third dose of any vaccine that comes along. The next thing to talk about is children, and so our schools have gone back. Kids have gone back to school largely. Not everyone has taken their kids back to school, and not all schools are back to a full complement. But our kids are back to school. But around the world, it's a big controversy about whether they should and where they should, particularly high schools and universities and colleges. So here it seems that important findings are that children are very unlikely to become severely ill, much less susceptible to severe disease. There is an unusual syndrome that got recognized fairly early on called multi-system inflammatory system syndrome that has infected a few hundred children around the world. And this particular syndrome, again, seems to be due to a specific immune response. And then, the other key issues, given that children are unlikely to become severely ill, do they nevertheless transmit the disease to others? And here it seems that children do carry the virus, can transmit it, but are much less likely to do so than older adults. The next story is about pre-existing immunity. So Griselda, in terms of your question, the really interesting thing is that 2-3 months ago, a study started to come out showing that up to 50% of people who were never exposed to SARS-CoV-2, as I said right at the outset, this is a new virus. And so we don't think that anyone until December or January of last year was exposed to this virus, nevertheless have T cells that can recognize it. So that's clear. And the second thing is, it seems clear that the reason some of us have these T cells is because human coronaviruses that cause the common cold are common around the world. And there are 4 varieties of these human coronaviruses. And that's why some of us have these activated T cells. The big question is, you have a T cell that recognizes the virus and it's shown to be so in the laboratory. We still don't really know whether these T cells have functional relevance. In other words, whether they give you sterilizing immunity or any other kind of immunity and protection from the virus. Now, on this slide, we're going to talk about the genetics of this. And there are 2 aspects to the genetics. One is the virus itself and its genome. That's the term for the DNA of a virus. And the second part of this is the host. So on this slide, I'm showing you to the left, the virus genome. And these linear arrays are individual viruses. At the top are alpha coronaviruses, a different kind of coronavirus. And the one we're worried about is a form of beta coronavirus. There are 2 viruses in that family or subfamily which cause SARS, one of which causes SARS, which was an epidemic a few years back, and MERS, a more recent one, that's the Middle East version of a severe disease caused by coronavirus. And then the arrow points to the coronavirus that we're experiencing today. And you can see those blue and orange bars are quite similar across the family, showing you that they are a family of viruses, but with specific differences. To the right is a family tree, or the technical term is a phylogeny. And what this diagram is showing you is that the virus does mutate and various strains are created. And although this looks like they may be very different, in fact, the mutations are conferring very small changes in general that are probably not causing huge differences in the kinds of disease that we're seeing or the kinds of problems in different countries. There may, in fact, be differences in the infectivity of the virus with a strain that's become dominant over the last few months. So there are two major strains in the world, and the one has displaced the other over the last few months. In terms of the human genome, so far there seem to be three major genetic variants in humans, so not in the virus, one of which determines ABO blood type that may have some effect, probably small, on who is susceptible to severe disease or not. There's another set of variants that seem to have to do with the receptor on human cells, where the virus attaches to our host cells and enters it to replicate. And then there's a third set of genetic variants that have to do with certain aspects of the immune response. So there are some genetic variants in human populations that may have some relationship to different ethnic or geographic populations that affect the host response. So once again, the immune system is important.
Sorry, Gary, if I can just interrupt you there quickly before you start going through the testing. Yeah, you've got your slide on antibody testing, so maybe you can just explain what that means, because I think there is some confusion about whether that means that you would be immune, or if that just shows that you've actually had COVID. And it was quite interesting when we ran the live event, we did a poll to see if people understood the probability of testing positive for COVID, depending on the sensitivity of this testing and the specificity of the testing. And we're actually quite surprised that people seem to understand that. Most of the people who answered the poll seem to get that question right. So you can just go through that example again. It was quite interesting.
Thanks, Isolde. So, yeah, I think people today anyway know the term antibody, and what's happened in the last couple of weeks is an antibody test was introduced. So this slide, in this slide a couple of weeks ago, we talked about how there had been a lot of controversy around antibody testing, and in fact in the mainstream media, there were arguments back and forth, mostly against antibody testing. So that headline about false hope. I think there was a big concern that antibody tests would be misused to diagnose COVID. So these tests became available 5-6 months ago, and were introduced sort of without regulatory approval, and misdiagnosing COVID, either that you had it or didn't have it, or were even potentially immune and protected from it, was a huge concern to the public health authorities in South Africa and in other countries. Nevertheless, SAHPRA, which is the regulator in South Africa, on August the 26th, or just a few days before that, announced that they had approved and issued regulations or guidelines for the use of antibody testing. So you can now go to a private lab, any of the major ones, and get an antibody test, which, as we said earlier on, is about B cell function and takes a couple of weeks before those antibodies are produced, but it's pretty reliable indication, we can say, that you have or have had the coronavirus. And the guidelines issued by SAHPRA are very clear about the intended use or recommended use of these tests. So firstly, they're for professional use only. You can only get them if they're prescribed for you by a doctor. The second major reason for using them is epidemiologic study. In other words, to try to understand how many of us have had the coronavirus or not had it. SAPRA has been very clear, and NICD has been very clear about the reliability of a positive test in terms of predicting immunity. So they're not a guarantee of immunity, and they're not diagnostic. And in other words, if you come down with symptoms that may suggest that you have COVID, you should not use an antibody test as a diagnosis or to support the diagnosis. So between the Foresight event and today, Griselda, a fascinating seroprevalence survey was announced or reported, which was done in the Western Cape, that showed a very high seroprevalence in specific population of the Western Cape in the Cape Town area. So they sampled, these were blood samples taken from HIV surveillance and from prenatal clinics. And somewhere around 37–40% of those samples turn out to have antibodies, suggesting that somewhere around 35–40% of that population have had the coronavirus, which is much higher than we would have thought, and much higher than I think practically any other seroprevalence survey that's been published in the world. The comments in the bottom right frame there, just to re-emphasize that although antibody tests can't be used ordinarily for testing, they do have a specific role. If you have somebody who seemed to have had the illness clinically but wasn't tested, it could be used to confirm that it was a COVID-19 in those people and in children who develop this rare multi-inflammatory syndrome. And Griselda, as you said, we had a very smart Foresight audience who got the question right. And the question came out of this slide. So this is an interactive calculator that you can find on the internet, published by the British Medical Journal. And this is kind of a scenario that is, I think, poorly understood even by many practicing clinicians, which is around the sensitivity and specificity of these tests. So the scenario here is that if you have a pre-test probability, that means what you think the prevalence or likelihood of the disease is in an individual that you're testing. So if we had someone without symptoms at all in an area of South Africa where we thought, well, maybe 5% of the population have had COVID, then if you had a test with a sensitivity of 70%, so that's this number here, and a specificity of 96%, what is the likelihood that a test that comes up positive is actually a true positive? So with a test that's as good as this, with a 96% specificity rate, as many as 50% of these patients would have a false positive result. And that is really the concern that the public health authorities had, that if our prevalence is low in the population, so if we used it for screening, a healthy population, half of those people would have a false positive result and draw a wrong conclusion from a test like this. If, on the other hand, we had a pre-test probability, so this is for screening, and in other words, testing people without symptoms, 20% and a slightly better test specificity, then we'd have a much more reliable result come out of it, where a positive test would mean a 93% probability that the test is, a positive test is a true positive. So our audience did rather well on the quiz that we presented them with on that particular day. And here what I want to emphasize is that it's not only the test result here that's important, but what you do in response to it. So if you use a test like this to actually diagnose and treat COVID, you may run into problems. If you use a test like this to quarantine patients and isolate them for 10 days, again, it has significant consequences, but possibly not as potentially harmful. And certainly if you're using it to admit someone to a COVID ward, you better be sure that your pretest probability is high and that your test sensitivity and specificity are really high as well. And here the idea of the immunity passport was really a bad idea that, again, the public health authorities in South Africa and elsewhere have tried to dissuade people from using. Okay, so one of the last things to talk about today is the idea of herd immunity. So we heard a world leader yesterday talk about herd immunity in a completely inappropriate way. Herd immunity refers to this idea that individuals who have had the virus or who are protected in some other way limit the spread. And if we get to around 70%, seems to be the magic number, 70% of the population that do not spread, are invulnerable, not susceptible to the infection, then the exponential, the pandemic, the epidemic essentially stops. That doesn't mean that the virus goes away. What then happens is the incidence or the prevalence of the disease day to day, month to month, drops to an endemic level, so it may still be around, but we escape the exponential rise and the massive impact on the healthcare system and on the country as a whole. So that's where we want to get to, herd immunity, and we can acquire it if we allow the virus to sweep right through the population, which is generally an undesirable way to go about things, as we know, or we can achieve it through a vaccine. And a vaccine is the safe way to achieve herd immunity, and it's much more effective, actually, with a good vaccine than it would be in allowing the infection to spread across the population. The last thing I want to talk about today, before we end, is something that people are referring to as long COVID, which is that COVID not only causes, in certain people, severe disease or even death, it may unfortunately persist. So people who have symptomatic but mild disease or more severe disease, certainly those people who end up in hospital, may recover fully, but a certain proportion of them, as many as perhaps 10 or 20%, have ongoing symptoms. So obviously at this stage we don't know exactly how long for, but there are more publications coming out to suggest that people have chronic fatigue, chest pain, shortness of breath, cognitive problems that are persisting and causing a lot of problems for these individuals. And at this point, we don't know whether it's due to perhaps residual damage caused by the virus and the infection, or whether, again, it's the immune system that's having ongoing activity that is causing these problems. So we'll discover over time which of those causes and how bad this is and how many people it affects. But again, it's another reason for us not to relax as the epidemic wanes or the pandemic wanes, but to be on guard and to continue to look for not only strengthening of the public health response, but for medicine and vaccine. Okay, so I'm going to summarize quickly for you. I think the major points to take home are firstly that the immune system is remarkable, complicated. It enabled us to live with, largely without problems. Occasionally they overcome us, and occasionally that set of circumstances which we have seen with COVID-19 is due to the immune system response rather than any direct effect of the organism secreting toxins or other ways that organisms like this can harm us. So I've simplified it in 2 or 3 slides. Essentially, we talked about the innate system, that initial response. Secondly, the adaptive, clever, targeted part of the immune system. And then finally, that the immune system leaves us with a memory of microorganisms and viruses that enables us to have a much more effective response the next time we see them. T cells are the key actors along with B cells in protecting us from COVID and other viral disorders. Immunity is a question of degrees, not absolutes. Hopefully we have sterilizing immunity. Sometimes we can be reinfected. Sometimes infection is incomplete. Hopefully many of us who have been exposed to cold viruses in the past will have protective immunity to SARS-CoV-2. Whether we become sick or are protected is a question of a balance in the immune system that allows for addressing the virus but not attacking our own host tissues, and the timing there is critical. So here it's interesting to think about the balance that we need in the immune system, which transfers perhaps to the idea of a balance in the policy response to COVID-19. So many people are starting to say that we've overreacted to the pandemic and damaged the economy. And of course, I'm not going to answer those questions or address those questions today. Certainly the virus has created huge uncertainty, not only for medicine, but for policy. But over time, certainly the answers will emerge about the biology of the virus and how to deal with it. I'm not sure that we'll have such clear answers about what the policy response should have been or should be going forward. But hopefully a better understanding of immunity and COVID-19 will be helpful for all of us going forward.
Thanks, Gary. Very interesting. Yeah, I think what I took away was just, noting the differences between the protective elements of your immune system, or the part of your immune system that's sterilizing, and the cytokine storm. So sometimes actually your own immune system causes the disease, which is interesting. A lot of what I took away from the presentation. So thank you.